Hello students, you are welcome to today's edition of this lecture. I'll be taking you through a quick tour on epidemiology. Starting with the study session 1, we have to talk about epidemiology, the basic concepts and definitions. Sit back and relax to enjoy this lecture. Thank you. Moving to the introduction, public health is primarily concerned with the prevention of diseases in human population. It differs from clinical medicine, both in its emphasis on prevention rather than treatment, and in its focus on populations rather than individual patients. At the end of this study session, you should be able to define the concept of epidemiology, which include population and determinants. You should be able to explain intrinsic determinants of diseases and discuss the extrinsic determinants of diseases. What is epidemiology? A useful general definition is that given by Squabel et al. In 1977, it defines epidemiology as the study of diseases in populations. It thus differs from more conventional medical approaches to the study of diseases that is normally concerned with the study of disease processes in affected individuals. While the objective of the latter is to find cures for diseases in individuals already affected, epidemiology is basically concerned with the reasons why those individuals became diseased in the first place. Inherent in the epidemiological approach is the belief that the frequency of occurrence of a disease in a population is governed by the interaction of a large number of different factors or determinants. Epidemiology has been defined by Last in 1988 as the study of the distribution and determinants of health-related states or events in specified populations and the application of this study to the control of health problems. A population can be defined as a complete collection of individuals that have some particular characteristics in common, depending on the characteristics being considered. A population can be very large or small. And when we are talking about population at risk, which is usually used in epidemiology, is a common term used in epidemiology, it refers to a subset of the originally defined population, and it comprises the total number of individuals in that original population that are considered capable of acquiring a particular disease characteristic or disease characteristic that is being studied at that particular time. For instance, we might be interested in studying the frequency with which abortion occurs in a population of dairy cattle of a certain breed in a certain country. The population at risk will not be all the individual animals of that particular dairy breed in that country, since this would include males, steers, and immature females, all of which will not or could not be pregnant and therefore could not abort. It will consist of female cattle of that breed, which were of breeding age. What is determinant? A determinant is any factor or variable that can affect the frequency with which a disease occurs in a population. Determinants can be broadly classified as either being intrinsic or extrinsic in nature. If we accept the premise that the frequency with which a disease occurs in a population is governed by a number of determinants, it would be expected that some of these, particularly the extrinsic ones, would vary in space and time. It follows, therefore, that disease is a dynamic process. Intrinsic determinants, they are the physical or physiological characteristics of the host or disease agents, which can be intermediate host or vector if present, and they are generally determined genetically. Looking at disease agents as determinants of diseases. Agents associated with disease can be categorized into two broad groups. We can have living agents such as viruses, bacteria, rickettsia, protozoa, helminths, anthropoids, and etc. We can have non-living agents too, such as heat and cold, water, nutrient toxic substances. In instances of infectious diseases, the presence or absence of the etiological agent is the main determining factor in the epidemiology of the disease. Obviously, disease cannot occur in the absence of the agent, but conversely, disease need not always result from the presence of the agent. There are various terms that are related to disease causation. We can have infection, which is defined as the invasion of a living organism, the host, by another living organism, which is the agent. Disease itself is defined as a derangement in the function of the whole body of the host or any of its parts. We can have infectivity, which is a measure of the ability of a disease agent to establish itself in the host. This term can be used quantitatively when an agent is referred to as being of low, medium, or high infectivity, or quantitatively. We can have various other terms too, including virulence. Virulence is defined as a measure of the severity of a disease that is caused by a specified agent. Virulence is usually used to measure the varying ability of disease agents to produce disease under controlled conditions. Another term is pathogenicity, which is an epidemiological term that is used to describe the ability of a particular disease agent of known virulence to produce disease in a range of hosts under a range of environmental conditions. Creation of the carrier state. The term carrier is used to describe an individual that is infected by a disease agent and is capable of disseminating that disease agent, but shows no signs of clinical diseases. We can have three types of carrier states. We can have a true carrier, we can have incubatory carrier, and we can have convalescent carrier. Convalescent carriers occur in such diseases as contagious bovine pneumonia. We can have the incubation period is the period of time that elapses from the infection of the host by the agent to the appearance of clinical symptoms. Let's look at the methods of transmitting infectious agents. We can have contact transmission. In contact transmissions, the agent is conveyed between hosts through direct physical contact, as in the case of venereally transmitted diseases such as fibrosis or trichomoniasis, or through indirect contact. In cases of indirect contact, the agent is normally contained in the excretions, secretions, or exhalations of the infected host, that is, in the feces, urine, milk, saliva, placenta, or placental fluids, or as aerosols on droplets in the breath. Susceptible hosts contract the infection either by direct exposure to these or through exposure to substances contaminated by them. Diseases spread in this fashion include rinderpest, foot and mouth diseases, Newcastle diseases, and contagious bovine pneumonia. The main factors determining whether or not transmission takes place in contact transmitted diseases are 1. The ability of the agent to survive in the environment. 2. The extent of the contact that occurs between infected and susceptible individuals of the host populations and their mobility within these populations. Looking at another means of transmission, we can have vehicular transmission. In vehicular transmission, the agent is transferred between infected and susceptible hosts by means of an inanimate substance or object, sometimes called fomite, such as water, foodstuff, bedding materials, veterinary equipment, and pharmaceuticals, or on the skin, ear, or mouth part of an animal. In contrast to indirect transmission, the survival time of the agent in or on the vehicle is usually prolonged. This means, in effect, that vehicular transmission can take place over greater distances and over longer time periods. Hygiene, disinfection, and control over the distribution of likely vehicles of transmission are important factors in the control of vehicular transmitted diseases. Let's look at host determinants. The main intrinsic determinant in the host, which can influence the frequency of occurrence of infection and diseases, are species, the breed, the age, and sex. The multi-species susceptibility to disease agent is particularly important if the species concerned are able to maintain the disease agent within their populations, that is, to function as natural reservoirs of infection. Differences in susceptibility to disease are often seen between different age groups. For example, young animals are generally less susceptible to tick-borne diseases than older animals. Looking at the extrinsic determinants of diseases, there are three major extrinsic determinants. The first two are the climate and soils, which by interacting in a variety of ways affect the environment of the host, the agent, and the intermediate host or vector. If they are present, the third major factor, apart from the first two, the climate and soils, is man, who uniquely among animals has the ability to modify the environment in which he lives and the environment in which he keeps his livestock. Thank you for listening. See you in the next study session. Bye for now.